I would like I would like to spend the next set of slides discussing probably something you're familiar with: eukaryotic cells. Eukaryotic cells are the ones that, again, you're more than likely been introduced to in middle school. These are your organized cells. In other words, eukaryotic cells are those cells that have what we are familiar with as all those parts. In middle school they may have introduced it as kind of like works like a factory, each part doing a specific job. And so we have this word eukaryote. Eu means true, karyote, cell. So true cells. These are your more complex cells. And again, the two slides that are coming up, the first one shows that animal cell. The animal cell, the most significant organelle in there is that nucleus, and it tends to have possibly a regular shape because the outer membrane is the cell membrane. There is no rigid outer structure giving it a regularity to the shape. And then the second cell is the plant cell, and the biggest significant difference there initially is again, a plant cell has that very thick outer, what we call cell wall, leading plant cells to have a regular, often rectangular or square shape. Another significant characteristic of plant cells is they have chloroplasts, giving them a green color. They also usually have a very large vacuole. That's due to the fact that the chloroplasts make sugars, and that vacuole stores these sugars for a significant period of time. Now, at this point, what I'd like to do is just run through some of the general characteristics of eukaryotic cells, trying to perhaps highlight what separates them from the prokaryotes that you learned about and heard about earlier. First, eukaryotes have a distinct nucleus. And again, this nucleus contains all of the cell's DNA, all the cell's information. And in eukaryotic cells, they evolved to have this cell nucleus in order to ensure that as the cell divided, all of those multiple strands of linear DNA would divide properly and be copied properly and be utilized properly. Unlike prokaryotes, there's usually multiple strands of DNA inside the nucleus, and we'll talk a little more about the nucleus later. Another characteristic of eukaryotic cells, which kind of lends themselves to why they get their name, is that they have elaborate internal membrane systems. Now, these membrane systems are what lead to or have led to the formation of most organelles. There are a few that may have entered the cell through as prokaryotes, and we'll talk, and become organelles later. We'll talk more about those. But the ability of these membranes to form allows the cell to increase efficiency. In some cases, the membranes will increase surface area to volume ratio within those organelles. It increases efficiency because it allows, oftentimes, these membranes the facilitation of proteins and other chemicals to move within the cell. And so these membrane systems are really important. Now, as part of these membrane systems, there are several strategies that cells utilize that I would just like to talk about a little bit here, that are fairly unique to eukaryotic cells. You might see these in both animal eukaryotic cells as well as in protista, animal-like, or protozoan cells. The first one that I'm going to talk a little bit about here is phagocytosis. I mentioned phagocytosis earlier when we talked about viruses. And the bottom line is, phagocytosis is the process whereby one cell alters its membrane, okay, in order to absorb, in order to ingest, okay, food sources, oftentimes bacteria, sometimes just various nutrients. The two pictures that I've selected to show you here, the first one shows an amoeba. And as you can see in that first slide, the amoeba is creating what are known as pseudopods. And those pseudopods are beginning to stretch out, and those pseudopods are beginning to envelop, okay, a food particle, which will then be absorbed through the cell membrane directly into that amoeba cell, where it can be broken down by digestive enzymes. The second one, okay, the color photo there, as the caption says, shows a white blood cell. That's the one on top, okay, and that white blood cell is in the process of devouring E. coli through the process of, again, phagocytosis. We had talked about this a little bit earlier, that we have these phagocytes, these variety of cells within our vascular system inside our blood vessels, which, again, are there to protect us by eating, absorbing foreign invaders. And so again, this first strategy here is this process known as phagocytosis. Now, the second strategy that I want to talk about is one where eukaryotic cells may form on their surface what are known as trichocysts. Trichocysts are basically microscopic poisonous darts. In the slide that I've shown you, you can see that those trichocysts are coming off the surface of a paramecium cell. The paramecium cell, the single cell paramecium, a protozoan, common freshwater protozoan, uses those trichocysts in order to basically immobilize its prey. And once it's been immobilized, that prey can then be ingested, swept down the gullet of the paramecium right into the cell for digestion at a later date. Okay. The third strategy that I want to talk about for a few minutes here, a moment or two, is called endocytosis. Now, in this strategy, okay, cells, as the word says, endo, into the cell. Endo, inside, cell, cyto, cell. And what happens in this strategy is materials, okay, sometimes viruses such as HIV will attach themselves to proteins that are on the cell membrane, and when they attach themselves to proteins on the cell membrane, the membrane will then, under a series of chemical changes, absorb those materials into the cell itself. Endocytosis plays a significant role as part of how your nervous system works. In your nervous system, chemicals are transmitted from one nerve cell to the other across a space, a fluid space called a synapse. Those chemicals will attach to the next nerve cell or neuron, and once they attach, they are absorbed and they trigger that neuron to carry an electrical message. And so this process is called endocytosis. The reverse of endocytosis is the process known as exocytosis. In this process, materials are pushed out of the cell through the cell membrane into the environment. Exocytosis is a common strategy for cells that are involved in the secretion of certain chemicals. Exocytosis, for example, is important in the cells of the salivary gland. they will secrete, okay, enzymes through the cells of the salivary gland into the gland itself, which are then released into the mouth, and these enzymes can then carry out, again, cell, okay, digestion, or begin the process of digestion of chemicals in the mouth. And so exocytosis, and this is, again, something that you see in both unicellular, multicellular organisms. Exocytosis occurs when hydrochloric acid and other enzymes are secreted into your stomach, or enzymes that are secreted into your small intestine to help with the process of digestion. And then a third strategy is known as pinocytosis, and this is basically when the cell alters its cell membrane, usually just altering those lipids, as we'll talk about in a later unit, a later section of notes. But basically, small particles of fluid are absorbed into the cell. And so pinocytosis is a bit rarer, but it's a process, again, of absorbing these fluids inside, okay, into the cell itself. So these are the essential kind of characteristics and strategies that you usually find in these eukaryotic or complex cells. Now, at this point, I want to kind of focus in on the variety of organelles that are found inside a eukaryotic cell. All right, we're going to start with that control center of the cell, the nucleus. Now, this small drawing that you see at the bottom here shows a nucleus, and one of the things that stands out when you see it is that the nucleus is like the only organelle within a cell that actually has another organelle within it. And we'll come back to that in just a little bit here, but that's important to recognize. Now, both of those components work together. As it says here, the function or the job of the nucleus is to store all the information that the cell needs for survival. Now, we haven't talked yet. We will spend a good chunk of this year looking at what we mean by genetic information. And the bottom line is, as you've heard various times throughout this year, genetic information really comes down to DNA. So all of the DNA for the cell is stored inside this nucleus. It doesn't ever leave the nucleus, okay? The only time it's ever freed from the nucleus is during cell division. Other than that, it's stored in here, and the cell will read that information and will pass information out of the nucleus when necessary for the cell to do a job. Now, the nucleolus is that organelle that's built into the nucleus itself. And the nucleolus, again, what it does is it plays a role in the formation of ribosomes. As we will talk about later, ribosomes are utilized for making proteins. And proteins are built from information, okay, on DNA. Ribosomes are actually made of a type of RNA, which we'll talk about in another unit. Now, again, this picture here gives you an electron microscopy picture of, a nucleus at about probably 6,000–7,000 power magnification, and you can see the nucleus in there. In this next one, you can see one of the things that stands out, as in again another picture of the electron microscope, is that there are pores, holes in the nucleus, and that's where this information, the pieces of DNA information, leaves the cell and goes out and travels, okay, into other parts of the cell so it can be utilized. Then we get to the mitochondria, the powerhouse of the cell. Now, the powerhouse of the cell gets that name because it does aerobic respiration. It carries out the process of respiration, making ATP molecules for the cell to utilize to do what jobs it needs to be done. You'll notice in this drawing, the inside of a mitochondria, okay, has all of these folds. Well, the question is, why are those folds so important? Those folds, again, increase that surface area to volume ratio, allowing more aerobic respiration to take place, and therefore more ATPs to be produced. One of the more interesting things about the mitochondria is, again, the fact that it has its own DNA. And this own DNA is what leads scientists, along with these chloroplasts, to understand that it is probably was once a prokaryotic cell that incorporated itself into a eukaryotic cell in order to enhance its ability to be able to survive. The next organelle I want to take a few minutes to talk about are the chloroplasts. Now again, chloroplasts come in a variety of appearances. The ones you're most familiar with are the green chloroplasts that harness the blue-violet areas of the spectrum. The purpose of chloroplasts is to carry out photosynthesis. One of the, again, unique things about chloroplasts is that you'll notice in this drawing here that they have internal components increasing, again, that surface area to volume ratio, maximizing the ability of these chloroplasts to carry out as much photosynthesis as possible, producing as much sugar as possible. And so again, it keeps coming back to something, surface area to volume ratio is an important concept in all of these cells in different ways. The last picture I want to just mention is, again, chloroplasts also have their own DNA. And scientists believe, again, that chloroplasts, like the mitochondria, were once prokaryotic cells who found it more successful to become a part of the eukaryotic cell. Scientists refer to this process as endosymbiosis, okay, the endosymbiotic theory. And scientists believe that there's strong evidence for this based on the characteristics of both these organelles, mitochondria and chloroplasts, as well as the fact that their own DNA gets copied by each organelle when the cell divides. All right, the next series of organelles that I want to talk about with you guys, they're all kind of linked together. We'll start with the ribosome. The ribosome is an extremely important organelle. We talked about it when we talked about prokaryotes. The ribosome is where all the proteins are made in the cell. This is where that DNA information stored in the nucleus is transferred so that the cell knows what to do with it. And it's inside the ribosome that these actual proteins are manufactured. So it's basically a ribosome. The ribosome is a protein factory. As you can see from this electron microscopy picture, the cell is filled with hundreds, thousands of these ribosomes. And again, the reason for so many of them is because ribosomes, proteins that are made by the ribosomes, are extremely valuable in so many different ways for a cell. The second organelle that's linked to these proteins is the endoplasmic reticulum. Now, the endoplasmic reticulum comes, well, basically the word endoplasmic, inside the fluid, reticulum, skeleton type structure inside the fluid of a cell. There are two kinds. Some are known as rough ER because they are covered with ribosomes along its surface, giving it under the microscope a rough appearance due to the presence of these ribosomes on their surface, and we'll come back to that in a minute. And then some are known as smooth ER, okay? And the reason, again, for this different adjectives, smooth ER versus the rough ER, is smooth ER does not have these ribosomes attached to their surface. You tend to find smooth ER in cells that are one of their functions is to secrete chemicals into their environment. So they use the excess endoplasmic reticulum, the smooth ER, to facilitate this process, which again leads into this What do? Well, their major job is to transport proteins throughout the cell. Again, remember, all these organelles are to improve efficiency in the cell. So the ER acts as kind of like a tube network to ensure that these proteins are moved wherever they need to throughout the cell, and they also support the cell. All right, the next organelle I want to talk about are the Golgi bodies. You may see several of these Golgi bodies inside the cell. Golgi bodies, okay, carry out several functions. One of their main functions is to package up proteins. And so they are kind of the third link in this protein connection, starting with the ribosomes, making proteins, the endoplasmic reticulum, moving proteins, Golgi bodies, packaging them up. And Golgi bodies will package them into these components called vesicles. Now, animal cells, at some point in their life cycle, will actually produce vesicles from their Golgi body called lysosomes. And we'll talk about those here in just a minute. But that's a secondary function. And so one of the key things to keep in mind is that, again, these Golgi bodies, okay, are the third link. Ribosomes make protein, endoplasmic reticulum, move protein, and Golgi bodies package protein. All right, let me talk a minute now about the lysosomes. The lysosomes, these vesicles / organelles that are produced through the Golgi bodies, primarily in animal cells, play a job of, they basically store enzymes to help the cell with the breakdown of important molecules that they need for nutrition. Another interesting aspect of the lysosome is that they actually play a role in killing the cell, destroying the cell, breaking down its components at the end of its life cycle. And so you can see when you look at these pictures of lysosomes, they come in a variety of names. Peroxisome, because it's got hydrogen peroxide, a very harmful, very toxic chemical that, again, breaks down materials very easily. They're very dark because they're very dense. And so these lysosomes, again, play a significant role in the manipulation, the digestion, the breakdown, the transfer, the chemical change of nutrition, as well as the breakdown of the cell at the end of the life cycle. And they're only found, again, in animals. The next organelle I want to talk about are the vacuoles. Every cell has vacuoles. And again, these vacuoles play a role in the storage of materials within the cell. They can store food, minerals. They can even package and store waste until the cell can remove them from the cell. In this first slide here, okay, you can see, again, under the microscope, you can see a picture of, on the left, some vacuoles that are being used for storage in an animal cell. These are frequently known as contractile vacuoles. And so when the cell needs to remove them through exocytosis, something we mentioned earlier in this slideshow, it will move this vacuole to the cell membrane and basically squeeze it through the cell membrane through that lipid barrier. On the right are plant cells, and you can see in those plant cells the large vacuoles. And that's one way you can oftentimes identify, again, a plant cell is by the fact that it's got extremely large, okay, vacuoles, often storing starches and sugars that are the product of photosynthesis. Continuing our overview of the organelles here, the next one I want to talk about for just a moment is the cell wall. Now, the cell wall, again, is one that's just found in plants, as opposed to those lysosomes mentioned earlier, only found in animal cells. And so it's a second organelle found solely in plants, along with the chloroplasts. What does the job of the cell wall do? Well, the primary job of the cell wall is to provide support for the plant, I mean, it's very thick, as you'll see in one of the future slides here. It also provides a very sturdy barrier to protect the cell from environmental changes, severe environmental changes in its environment. And so the cell wall made of this cellulose provides the ability of the cell to support itself. Also provides a barrier to allow the cell to withstand severe environmental changes. All right. The last organelle I'd like to talk about for a moment here is the centrosomes or centrioles. We'll come back and look at these in actually the next unit when we talk about cell division. But these guys are found in animal cells primarily, and they create the spindle fibers, a protein type structure, which is used only during cell division. They are used to manipulate or move the DNA to various parts of the cell, so the cell can carry out cell division in a manner that allows the cell to divide the DNA equally up, appropriately up between the original parent cell and the two daughter cells. And we'll see the role of this when we get to the next unit. In closing, let me just kind of review these organelles briefly, highlighting again the fact that many of them are found in both plant and animal cell. But one of the things to understand and to recognize is that there are a few that are unique to only animal cells, and those are the lysosomes and those centrioles that we'll see in the next unit. Okay? And so it's important to make a note of this in your notebook, again, that these two are unique to animal cells. And again, as we can also look at in the next slide that's going to show up here of a plant cell, many of the same organelles are also present here in plant cells that are found in animal cells, thus the common ancestry of both these eukaryotes and such, both plant and animal eukaryotic cells. But the two organelles that are unique to plant cells are the chloroplasts and that cell wall. And so important to make a note of that as well.